Hook 30 sec. What if I told you that we can now predict how proteins fold, with 90% of their structure hidden? That's exactly what a team of researchers has just done. They built an AI that looks at only 10% of a protein's 3D shape and reconstructs the rest. And it's not just a neat trick. It's revolutionizing how we understand the building blocks of life. But here's the question. If an AI can do that, what else can it learn? Welcome back to the channel. Today, we're diving into a paper that dropped on May 20th, 2026, literally yesterday. And it's a big deal for anyone who cares about protein science, AI, or drug discovery. We're going to talk about a massive new benchmark called TED bench and a method called masked invariant autoencoders, MIAE for short, that's setting new records. By the end of this video, you'll understand why protein folding AI is about to get a whole lot more powerful, and what that means for the future of medicine and biotechnology. Stay until the end, because I'm going to tell you why 90% of a protein might be all you need to see. Act 1, the problem. Let's start with a fundamental question. How do we teach AI to understand proteins? Proteins are the nanomachines of life. They fold into complex three-dimensional shapes that determine what they do. If you know a protein's shape, you can start to understand how it functions, how it interacts with drugs, and how mutations cause disease. Now, for decades, the gold standard was experimental structures, proteins that had been painstakingly mapped using X-ray crystallography, cryo-EM, or NMR. But those are expensive and slow. We only have about 200,000 experimentally determined protein structures in the protein data bank. Compare that to the hundreds of millions of known protein sequences, and you see the problem. Then AlphaFold came along and changed everything. Google DeepMind's AI predicted protein structures for nearly every protein in the known universe. Suddenly, we went from having structures for a tiny fraction of proteins to having predicted structures for hundreds of millions. But here's the thing, predicted structures aren't perfect. They have blind spots, they can miss conformational changes, and they might not capture the full complexity of protein dynamics. So the research community faced two challenges. First, how do we build a benchmark that's big enough to train modern AI models, but also high quality enough to be meaningful? Previous benchmarks like SCOPE and CATH are great, but they're small. We're talking maybe 10, 000 to 20, 000 structures. That's not enough for the kind of deep learning we're doing now. Second, how do we learn useful representations from all these predicted structures without needing labeled data? Because labels, like knowing what fold a protein belongs to, require expert annotation, and that's expensive. Here's what you're going to learn today. First, why the old benchmarks weren't cutting it. Second, how researchers built TED bench, a massive new benchmark for protein fold classification. And third, how they created a new self-supervised learning method called masked invariant autoencoders that absolutely crushes the competition. Stay until the end, because I'm going to tell you why this matters for the future of drug discovery, protein engineering, and our understanding of the protein universe, and why 90% masking is the secret sauce. Act 1, the problem. Here's the problem. Protein fold classification is foundational to structural biology. A protein's fold, the way its amino acid chain arranges into three-dimensional space, determines its function. If you can classify a protein's fold, you can start to infer what it does. But the protein universe is vast. There are estimated to be hundreds of millions of protein sequences, and we only have experimental structures for a tiny fraction, about 200,000 in the protein data bank. Now, AlphaFold changed the game. We now have predicted structures for hundreds